Hello, Mrs. H here. We are going to start with a couple of easier fact recall questions, and then we're going to get really stuck into some more difficult application questions while going over some common exam techniques. Hot flushes are one of the symptoms associated with the menopause. These symptoms are thought to be linked to falling estrogen levels. Low levels of estrogen are thought to disrupt the thermoregulatory centre in the brain. Name the part of the nervous system which includes the brain and state the exact location of the thermoregulatory centre. The part of the nervous system is the central nervous system and the exact location is the hypothalamus. So nice bit of recall there. MRI scans have been used to investigate the structure and function of the human brain. Scans are essential when health professionals suspect that a stroke has occurred. This is an MRI scan of the brain. Identify the following structures, letter B and C. So B is the cerebellum and C is the medulla oblongata, and of course A is the cerebrum or cerebral cortex, and there your D is the top of the spinal cord. The image below is a diagram of the human brain. Which of the labelled regions would be directly involved in learning to play a musical instrument? Well, that learning is going to need to use the cerebrum or the cerebral cortex and also the cerebellum. So those parts are Z and W. So the answer is C. The amplitude and duration of the action potentials in a single neuron are always the same. However, the amplitude and duration of action potentials can vary between different types of neuron. Figure 2.3 shows action potentials in two different types of neuron. So if we just enlarge this a little bit so we can have a look. When you see these graphs, you know they are to do with action potentials, and that when the membrane potential becomes more positive, less negative, then that is depolarization. You know that when it returns back down to more negative, that is repolarization. Then when the membrane potential is lower than the resting potential, that is hyperpolarization. And then eventually it gets back to resting potential. Very quickly it gets back to resting potential. Now those are the key terms you need to use in your answer. So then you've got to think about, well, why does depolarization happen? What causes the charge to become more positive? And then you remember, it's the voltage gated sodium ion channels that are opening, allowing sodium ions to diffuse into the axon, depolarization. And then you need to remember that the repolarization is happening because the voltage gated potassium ion channels are opening, letting out the potassium ions. The voltage gated sodium ion channels are closing. So inside the neuron is becoming more negative. So it's repolarizing. And then we have that short period of time where the membrane potential is lower than resting potential, which is the hyperpolarization. Another thing that you might notice here is that the resting potential for the Purkinje neuron is actually more negative than for the dopamine neuron. So the dopamine neuron has a less negative or a more positive resting potential. So having looked at that graph, you now know the key terminology you need to make sure you include in your answer to any question on this. So what is the question? Describe the differences between the two action potentials and suggest explanations for the differences you have described. So they're not expecting you to know the answer, but it has to be an educated suggestion. So we could write something like, the resting potential in a Purkinje neuron is approximately minus 80 millivolts. Difficult to tell from that graph, but it's round about there. It's more negative than the resting potential of a dopamine neuron, which is approximately minus 50 millivolts from that graph. This could be because a Purkinje neuron may have more sodium-potassium pumps in their membranes, therefore pumping out more sodium ions, leaving the inside of the neuron more negative, or the membrane potential more negative. Depolarization in a Purkinje neuron is much faster and reaches a more positive action potential peak above plus 20 millivolts compared to the dopamine action potential peak, which is below 20 millivolts. So this could be because there may be more voltage-gated sodium channels in the Purkinje neuron membrane, which would enable more sodium ions to diffuse into the neuron in a set time. This would also help to create a steeper concentration gradient within the axon, therefore stimulating the subsequent action potential sooner. There is no obvious refractory period in a dopamine neuron. Hyperpolarization lasts a long time and resting potential takes longer to be re-established. Now, possibly that could be due to there being fewer sodium-potassium pumps in the membranes of the dopamine neurons. The table below shows the membrane potentials of different neurons at a cholinergic synapse. So these are the synapses that use acetylcholine as their neurotransmitters. The data were recorded on 5 separate occasions as shown in the 5 rows. So if we have a look at this table, we really need to just look at these columns. So we've got 5 replicates. We've got presynaptic neuron A, presynaptic neuron B, presynaptic neuron C, and postsynaptic neuron. Okay, so we could draw a line to divide those pre, and post. And we've got 3 different neurons, A, B, and C, joining up with our 1 postsynaptic neuron. Now, if we have a look across row 1, so the first go, we have got an action potential in presynaptic neuron A, but not in B or C. So that doesn't create any action potential in postsynaptic neuron. Neither does row 2, 3, 4, but 5 does. And then if you look at 5, you can see that you've got an action potential in all 3 of those presynaptic neurons. So this is an example of summation where you have these 3 different neurons causing an action potential in the postsynaptic neuron. Now that is spatial summation. So that answer is C. The proteins in skeletal muscle myofibrils are arranged in groups to form a repeating pattern. What repeating patterns do you know that you see in muscles? Well, that is going to be a sarcomere. As the muscle contracts, the pattern changes. Describe how the pattern within a myofibril changes when skeletal muscle contracts. What I would do here is sketch out a sarcomere to remind myself of the different bits, and then I can access the question more easily. So if you remember, we've got our actin and then our myosin in the middle, and the myosin heads will pull that actin over it. We've got, this is one sarcomere here, we've got our Z lines that mark the ends of one sarcomere. We've got the A band, which is just the width of the myosin, so that's not going to change at all. And we've got the I band, which is the part where there's just actin. So the Z lines move closer to each other as the sarcomere contracts or shortens, and the I band and the H zone, so the H zone is this area where there's no actin in it, they are both going to shorten and decrease in size, and the A band will stay the same length. Which of the options A to D correctly describes how an endotherm would respond to an increase in temperature? A lovely, lovely easy question for you right now. That's dilation of arterioles near the surface of the skin. Erectile muscles contract, causing hair. No. Rapid contraction. No. Sweat glands release less sweat. Definitely not. So it's definitely A. Ice fish live in very cold water. Ice fish contain biological molecules that allow them to tolerate cold temperatures. A group of students investigated the effect of temperature on the activity of two forms of trypsin, human trypsin and ice fish trypsin. Part of their method is shown below. So use 10 centimeters cubed of 5% trypsin solution for all trials. Measure the enzyme activity at 10, 20, 30, 40 and 50 degrees C for both enzymes. Carry out each trial in the same pH buffer. Repeat the experiment 5 times at each temperature. Measure enzyme activity by recording the area of gelatin on a photographic film that is broken down over a set period of time, and then calculate the rate of enzyme activity at each temperature. Suggest and explain 2 improvements that would increase the validity of the student's investigation. When you see the word valid, you know that means how well does the investigation measure what it sets out to. Well, you need to make sure that you're a bit robotic. When you see the word valid, you need to think, what are your control variables? Are there enough repeats? And is the range of the independent variable enough? So those are the 3 things you need to check if you're asked to design a valid investigation or look whether something is a valid investigation. Make sure you're checking those things. Of course, to check the control variables, you need to be very aware of what the independent variable is and what the dependent variable is, so you know which variables you've got to keep the same. So let's have a look at the method. We'll use 10 centimeters cubed of 5% trypsin. That's a very clear control variable. Excellent. Enzyme activity 10, 20, 30, 40, 50 degrees C. Yep, that is good. But we need to make sure that we use a thermostatically controlled water bath for those temperatures. Carry out the trial in the same pH. It doesn't tell us which pH buffer. Possible limitation there. Repeat the experiment 5 times. Yep, good. Repeats. Lovely. Measure the enzyme activity by recording the area of gelatin on a photographic film that is broken down over a set time. Now, this needs to be standardized. So what we mean by that is we need to make sure this is done in exactly the same way for each repeat. Let's have a look at these temperatures again. What is the point of using such high temperatures? You've got 50 degrees C there, and we know that human enzymes are denatured above 40, and we're comparing human trypsin with ice fish trypsin, and ice fish live in very cold water, so they are definitely not going to be living in any water that is above 30 degrees C. So what is the point in investigating temperatures up that high? We could actually investigate lower temperature than 10 degrees C. And also we could investigate smaller intervals between the temperatures as well. So that will help us to get a more accurate optimum temperature. Shall we write our answers down? We could use the same area of film for each repeat. And that will make sure that temperature, which is our independent variable, is the only thing affecting the results. Another thing we could do is use temperatures at smaller intervals. So maybe go up in 2 degrees C. And this enables you to have a more accurate idea of what the optimum temperature is for each enzyme. So say, for example, we investigated and found our optimum temperature was between 10 degrees C and 20 degrees C. We don't know if it's 11, 12, 13, 14, 15, 16, so on and so on. But if we were testing temperatures of smaller intervals, and say the optimum fell between, for ice fish this would be, between 2 degrees C and 4 degrees C, then it's a bit easier to pinpoint our optimum temperature. So it would make it more accurate. Suggest appropriate units to use to represent the rate of enzyme activity in this investigation. The students find this quite hard, but you need to go back to the method and see what you have to do. Now, the 1, 2, 3, 4, 5th bullet point down, measure enzyme activity by recording the area. So that's probably going to be centimeters squared or millimeters squared on a photographic film that is broken down over a set time period. Time could either be seconds or minutes. So basically, you're just doing area over time, and that's how you present your units. You could have millimeters squared per second, or centimeters squared per second, or millimeters squared per minute, centimeters squared per minute. Or you could even write this as millimeters squared with the / and s, but that is the old format. They do prefer you to do the one I've put the brackets around. So if you could stick to that one, that would be better. The students recorded the temperature that produced the fastest reaction rate in each of the 5 replicates. These results are shown in the table 3. Lovely, great. Okay, it's always worth just checking. We've got mean, mode and median, because of course the word average is very naughty. Most of the time in biology we use the mean, but here we've got our 3 different types of average. One of the students made the following statement: I think the mean is more accurate measure than the median or mode for these results. Evaluate the student's statement. Now, whenever you see evaluate, in this example, you need to say yes and no. You need to say both of those. Sometimes you get other evaluate questions, which is pros and cons. You need to do both sides of the argument. Agree, disagree. Even if you don't disagree, or even if you don't agree, you still have to write both parts, or you won't get the full marks. So let's have a look at the data then. Well, there are 2 mode values for ice fish trypsin. So we could say that the mean is a more accurate measure than the mode, because there are 2 modes for ice fish trypsin. However, no, because for human trypsin, in the temperature of 10 degrees C is anomalous, but that has actually been included in the mean calculation. So there you go, you've got your yes and your no answer. The students wanted to know whether there was a difference between the reaction rates of the 2 forms of trypsin at 30 degrees C. They performed a statistical test on the mean of the 5 replicates for human trypsin and the 5 replicates for ice fish trypsin. Suggest the most appropriate statistical test for the students to use and explain why this test is appropriate. We're being asked to compare 2 means, and comparing 2 means is the student's t-test, and that's our first bit of the answer. So student's t-test because 2 means are being compared, and this is actually an unpaired t-test because the data sets are unrelated. In other words, the human trypsin is not from the same organism as the ice fish trypsin. So unpaired student's t-test. And that is it. Thanks for watching. I hope you found it useful. If you did, don't forget to like and subscribe for more content.